It's a great pleasure to be discussing some of the results, research we've been doing to assess the impact of influenza vaccination on the variability or the genetic variability of influenza in piglets. So as you heard from Dr. Murray, influenza is a very complex and notorious disease. It's an RNA virus, but it can be effectively controlled by vaccination. Vaccination has been very effective in reducing the lesions and the transmission rates, and for the last half a century or so, it has been thus the cornerstone of influenza control. Despite this, influenza is a major threat to the industry still because it's capable of evolving very rapidly, giving rise to new or novel strains and reassortants, which actually defeats the vaccines which are currently in use. So therefore, there is a continuous need for newer vaccines to keep controlling these viruses. So to maximize the profit of the swine industry, it's essential to go and assess all the risk factors that are associated with the introduction of a new virus or, just introduction of a new virus into the system. So many factors have been assessed and evaluated, but one of the most important questions that remains unanswered is, does or can influenza vaccination drive genetic diversity of influenza in piglets under the current industry conditions? To answer these questions, we initiated an investigation in which we analyzed 9,900 nasal swabs from the piglets. These samples were collected over a period of 6 months from 52 farms from 7 different states. So the treatments we assessed here were no vaccination, mass vaccination, pre-Pharaoh vaccination, and it's important to keep in mind that the farms were vaccinated with both a commercial or an autogenous vaccine. So to begin with, what we did was we screened all these 9,090 samples to assess for the presence of influenza by real-time PCR, and which ended up in the identification of 23 farms which were positive for influenza, which formed the actual basis for our investigation. And we selected 391 samples from these 23 farms for further analysis. Little background on how we selected these samples. We attempted to select at least 8 samples per farm per month, and if the number of samples was less than 10 per farm, then we went ahead and sequenced all the samples. The samples were chosen randomly within the farm, and we had samples with CT values all the way ranging from 16 to 35.57. And what we did next was to isolate the total RNA from the nasal swabs directly, and then amplified the whole genome, that's the IAV whole genome, using a universal set of primers, and then clean them up and submitted for sequencing using a high-seq rapid chemistry platform. All the sequences which passed the quality control were then subjected to de novo analysis and reference genome analysis, and then the sequences of the segments which were derived were annotated for functionality using FLAN annotation tool. And then finally, we used all these functional sequences for actually assessing the impact of vaccination on variability by a statistical tool called as AMOVA, which stands for Association of Molecular Variance. So the results, we were able to identify multiple subtypes in the farms and in the samples. About 97% of the farms exhibited more than one strain in the farms. There was only one farm in which we were able to detect a single strain. We were also able to co-detect more than one strain in 55% of the samples. And you can see from this graph, the first arrow closer to the y-axis indicates the co-circulation co-detection of multiple subtypes. About 101 samples contained the subtypes H1, H3, and N2. The next one actually refers to 95 samples. That means 95% of the samples contain a mix of H1, H3, N1, and N2, and 17 samples had a mix of H1, N1, and N2, and 12 samples had a mix of H3, N1, and N2. So then we moved on to see how this IAV is distributed in a farm. The columns indicate the months, the time points in which the samples were collected, and the rows indicate the farms that were investigated. PF stands for P for vaccination, and M stands for mass vaccination, and NV stands for no vaccination. So each and every cell that's colored here pretty much refers to a sample that was collected for that particular farm at the time point. The light green colored cells indicate that the farms were negative for influenza at that particular time for the farm, while all the other colored cells represent that influenza was present. The most important point here is the cells which are represented in purple color indicates a co-circulation of multiple subtypes. Purple here represents H1, H3, N1, and N2 combination, and yellow here represents a combination of H1, H3, and N2. So the take-home message is that whether or not a farm is vaccinated, it's possible to identify multiple subtypes in the farm over a period. So then we did quite a bit of phylogenetic to understand the variability underlying these sequences, to see how variable they are within a farm and between the farms. It's kind of little condensed or hard to see, but actually reveals the complexity and diversity behind this antigenic segment H1. You can see from this slide that at least we were able to identify 3 different clades, delta 1, delta 2, and gamma, and also multiple clades. So you can see a tremendous variability between the farms. I've actually tried to color code the farms using different colors, so you can actually see how variable they are within the farms and if there is a new introduction. We kind of saw the same trend with another fragment, that's H3. In this case, we were able to identify 4 different clusters, and we also see multiple subclades. We did this analysis for all the segments, but I'm not going to talk about them today. So moving on to the acid test, that's the analysis of molecular variation. So for those of you not familiar with this, AMOVA is basically a statistical model or a tool for assessing the molecular variation within a single species. It estimates the population differentiation directly from molecular data by testing hypotheses such as differentiation. Or in very simple terms, it's just an ANOVA analysis to analyze the variability in a given set of molecular data. So this can be done or elucidated from both the sequence data or the phylogenetic tree, but in our case, we use the sequence data. Basically, all the functional sequences were evaluated. We used a program called SPOPPER, which stands for an R-based program for population genetics, and this was developed by Commer et al. In 2014. We actually did this analysis at both the nucleotide and the amino acid level. We used the scripts that were available from this paper for analyzing the nucleotide, but we actually came out with our own script to analyze the data for the amino acid level. So we did this AMOVA analysis for all the 8 segments differently, both at the nucleotide level and at the amino acid level. And here is the summary of the p-values which we came out. We did 2 different models. The first model, we analyzed all the components in the investigation. That is, we took into consideration the samples, the farms, and the treatments. And you can see here very clearly that the variation between the samples and the farms were highly significant. Surprisingly, the treatments were not significant. You can see them in the red fonts. Very high p-values and nowhere close to even a suspect level, and which made us think that maybe there is something in the farm which is actually masking or confounding the results. So we did another set of analysis in which we removed the farm effect and did the analysis, and then we saw that the effect of the variation within the treatment actually became significant. We did the same analysis on the amino acids and pretty much came out with the same result or exactly the same results. So moving on to the conclusions, based on this study, vaccination does not appear to drive IAV diversity or variability in the herds investigated. Instead, IAV diversity in piglets appears to be influenced by other unknown factors and cannot be explained by the vaccination alone. We've seen tremendous amount of influenza genetic variability with multiple strains co-circulating within the herds. And the implications, vaccine use is unlikely to be a major driver of influenza virus change from the study, and most diversity appears to be due to co-circulation of different viruses, possibly introductions. And so in a nutshell, I think, or we think efforts should be made to decrease the introduction of new flu viruses in the pigs. Last but not the least, we'd like to thank our funding agency, USDA NIFA, for making this research possible. The PI of our research, Dr. Montse Thorimorel, co-PIs, Dr. Andres Perez, Marie Callahan, Srinan Srivatsan, Matt Allerson, and Dr. Alicia and Michelle Keeney. A special thanks to Fabian Chamba, Todd Knudsen, Anders Dias, and Samuel Hong, and Mai Ang for the help in the lab and their suggestions.